All right, well, it is 5:30 right now, and I know that a lot of people are still trickling in, but I would love to start on time. So, hi, everybody. My name is Corey Slack. I'm the Associate Director of Alumni Engagement. Welcome. Thank you so much for being here. We are so excited that you are out here, especially with all these tough times on what's going on in the world and uncertainty of what's going on as well. But we are so excited that you guys are here tonight and participating on this Zoom webinar. This is our very first speakers series with talking about COVID-19. And we are so excited to have Candace on here tonight to talk about all the things that she's doing with her team and what CSU is doing as far as research as well. I want to thank everybody that's participating. Tonight we have around 77 people registered. And if you guys don't mind just taking a quick moment, put in the chat your name, where you're from, what year you graduated. It's always nice to see where we're getting people from and even seeing what time it is in certain areas too. So take a minute while I'm talking and you guys can post some stuff in the chat as well. I would like to do a huge shout out to all of our members that are here tonight. We are so excited to have you. Out of everybody that's registered and here, we have a total of 26 members. We also have 11 lifetime members and then 5 grad pack members that are here. And grad pack members, for those of you who don't know, are members that have graduated recently in the past couple of years. So we are so excited that we have you guys here tonight. Without you, events like this would not be possible. And later on, I'll post something in the chat that talks about if you ever want to hear more about membership, you can click on that link and it'll talk about if you can be a member or how you can pay to be a member as well. I would love to throw it out to thank all of our university partners for tonight. The first one is Women and Philanthropy. We are so excited to be able to partner with such a great community that coincides with the goals of CSU's University of Advancement Guidelines. We would also love to thank the Office of Vice President for Research as well, helping us identify great speakers for this program and for this series. And then finally, the College of Veterinary Medicine and Biomechanical Sciences. We are so excited to be able to partner with every single one of these groups on campus and off campus as well. I will throw more information about each of those departments into the chat as well. So if you ever want to hear about what else we are doing on campus, research, anything that's going on, feel free to click on one of those links. I will do it as soon as I'm done talking and then once more before we end for the night. A couple more things. I will also throw into the chat my contact information. So if you're having any technical difficulties or having an issue with anything, please feel free to call me on my cell phone or email me. I'll be watching that as much as possible. We will have moments for you guys to answer or ask questions. We have a Q&A box as well as put your questions in the chat. I'll be monitoring them and then telling them to Candace as they come in. And on that note, I am so excited to introduce our speaker for tonight. Candace Mathiason is the director of CSU's Infectious Disease Research and Response Network team. Since her time here at CSU in 2019, she has been in the development of interdisciplinary teams to address infectious disease research from a broad platform. Each department and individual adds a level of diversity to the project that will lead to a better holistic understanding of the work that we do today. Candace is also a proud member of the Women and Philanthropy community. And without further ado, I will pass it over to Candace.
Thanks, Corey. Let's see. Here we go. I'm here. Yay. Thank you. I am really excited to be here this evening to share with you the work that I'll call it the IDRN, the Infectious Disease Research and Response Network, is doing here on campus, and in particular the work that we're doing on COVID. And so I'm going to start by sharing my screen with you. Let's see. Can everybody see that? I'm going to presume yes. And so as Corey mentioned, please feel free to put some questions in the chat box if you have questions as I'm going along. But I just want to give you more of a global perspective that will connect with many of the other speakers that you're going to hear in the series over the rest of this semester. So I'm excited to be able to provide that information just on a larger platform. So the Infectious Disease Research and Response Network, I will likely call it IDRN or IDRN for short. And the work that we've been doing to respond to COVID-19 has been pretty remarkable. So the team that has developed has submitted more than $33 million worth of funding opportunities. And so they're a very active group that are ongoing right now. So where did it come from? Where did this group initiate from? And as Corey mentioned before, the Office of Vice President for Research brought together this series a bit in identifying people that have been responding to the COVID pandemic. And so this group of people were part of the Office of Vice President for Research, or the VPR's Programs for Research and Scholarly Excellence. And so these PRSCs were established several years ago, and the IDRN itself was established in 2016. And what we did was combine a variety of individuals, some long-standing pillars within the Department of Microbiology, Immunology, and Pathology within the College of Veterinary Medicine and Biomedical Sciences. So who are we? So we started with about 60 faculty members that were part of this group, and the pillars included the arthropod-borne and infectious disease laboratories, or AIDL, if you are familiar with those groups, the mycobacteria research laboratories, the Prion Research Center, and the veterinary diagnostic laboratories. And so those were the initial groups that came together, and it was the real strength of the breadth of understanding of these major pillars in their research of infectious disease and the service toward infectious disease here at CSU that brought us together as a group. And since that time, we've now expanded. Over the past year, we've expanded to about 100 faculty members that connect 3 colleges, 11 departments, 4 centers, and Google. And you can see we stretch across campus, pretty heavy in CVMBS, where we initiated, but are making our way across campus to bring in other perspectives, again, to develop that interdisciplinary theme. So what is our mission? Our mission has been to address local, national, and international needs for infectious disease control and prevention. And really the goal is to promote, again, the development of interdisciplinary teams that can investigate, study, and develop mitigation strategies for diseases overall. So what has the IDRN been doing? Well, we've had a couple symposiums. One in 2018 here at CSU, and it was looking at metabolism of infectious diseases. And so local, national, and international folks came here to CSU to present their work, to develop collaborations, and then to take that information back out into the world. And so we've had a couple of really successful symposiums. I think we've had over 300 people at this symposium and at the second symposium, which was last summer, in which we, again, brought people from around the world to have a symposium on infectious disease diagnostics for the 21st century. So what do we have for diagnostics now that can detect diseases? And what's the future bring? What are new innovative approaches that we can be using to better detect diseases in the future? So we have those symposiums. We also are very active in promoting summer student fellowships. And so over the last couple of years, we've had 4 students that we have continued to support for their summer work. So these would be faculty within the IDRN and students that wouldn't have the opportunity to continue their research into the summer. But with IDRN support, they've been able to move forward. So again, over the past year, Corey mentioned that I've been the director for I guess since May of last year, 2019. And I'm really interested in how to bring folks together to start thinking on a more broad platform. So can we bring folks together and have fluid movement between interdisciplinary teams that would be able to bring their expertise not only to one team, but to use that information across a bunch of different teams as well. And so in the past year, we've developed 4 teams. The first being companion animal cognitive disorders. So think of diseases in humans like Alzheimer's and Parkinson's disease, other cognitive disorders. Can we better understand how those diseases work in companion animals, in this case, animals, cats and dogs, and be able to detect these diseases early, find therapeutics and ways to mitigate the disease earlier in the disease process. And so this is the first team that was developed through the IDRN starting last summer. There's another group that want to use equipment that we already typically use in the laboratory, so mass spectrometry, and use that information or use that equipment to better understand how diseases progress. Can we better understand the immunological approach that these diseases take to be able to get into a host and replicate within a host so that we, again, can address mitigation strategies for that. There's a third group that is interested in the role of the prion protein in cancer. So many of you may have heard of prion diseases. Chronic wasting disease is a prion disease here in Colorado that has spread across the United States and now into Europe and Asia as well. So that disease, as well as variant Creutzfeldt-Jakob disease or mad cow disease. Mad cow disease is a disease in cattle. Variant Creutzfeldt-Jakob disease is thought to be transmitted to humans through the consumption or blood transmission from people that consumed cattle. And so there's a normal protein that all mammals have. What this group is trying to better understand is whether or not that normal protein actually has implications in the development of cancer. And then, of course, what I will spend much of my time talking about this evening is the team that has come together to address the current pandemic, COVID-19 or the SARS-CoV-2. So the first group, just to give you some idea of how these groups have come together, the first group is IDT1 is being led by Dr. Julie Moreno. It's 14 faculty that are linking 3 colleges, 5 departments, and 2 centers in, again, using the diseases that occur in cats and dogs to better understand whether or not we can mitigate human cognitive disorders. The second group come together, Dr. Nicole Garcia, 4 faculty representing 2 departments and 1 center. Nicole actually works with mass spec, and the other individuals have come on board trying to determine in particular whether or not they can use this for COVID-19 to be able to better understand the disease process that's happening in the immune system to be able to better mitigate the disease. The third group, the role of the prion protein in cancer. Dr. Dan Regan is leading that group. He is a cancer biologist. 7 faculty connecting 2 colleges, 3 departments, and 1 center are working on this. And then, of course, the group that I will, of course, spend the most time on this evening is the COVID-19 group. Again, Dr. Alan Rudolph, he is our Vice President for Research, offered this opportunity to come together for a forum that was developed for both the community here at CSU as well as the Northern Colorado community for folks to come in and to be able to have their questions answered by a panel of experts in the field. And so Drs. Geis, Kading, Schounts, and Stallones were there. And this was in February, in mid-February, when we were first realizing that COVID was going to be spreading much further than we thought, and there was concern here in Fort Collins. So here's this team of individuals. We have 50 faculty that are bridging 3 colleges, 11 departments, 3 centers in Google that have come together to collaborate with one another And so we've sort of been matchmaking groups, people within this group, and they've been writing for funding. And as I've mentioned, they're well over $30 million in submissions for funding right now, and are having a really huge impact here in the United States, or globally, quite honestly, in the work that they've been doing. And I'll show you a bit of that in the next few minutes here. So within this group, there are 25 teams. They are studying things from one of the very first projects that took off was to better understand surveillance in humans. And so a team of individuals got together to try to better understand whether the retirement communities here in Fort Collins, could we surveil individuals, knowing that older populations were actually having the largest problem very early in the disease process back in February and March. That's really where we were seeing most of the COVID disease, as many of you have already heard this over and over, I'm sure. And so they got together to look at testing to see whether or not individuals within the retirement community, including the healthcare workers, had COVID, and whether or not they had infectious disease. And in fact, that's where we first learned that there was a lot of asymptomatic transmission. So somewhere between 40 and 50% of individuals were asymptomatic, but in the testing that was done, it was determined they still had an infectious virus, so could transmit the disease. We have a couple of researchers here on campus that look at bats and reservoir host development to try to better understand how did the disease transmit from potentially bats or civet cats into humans. There's a group that are also working on antiviral and disinfectant assessments, antiseptic wipes, and the development of hand sanitizer that's being produced here in Fort Collins and distributed locally, both to businesses and to campus itself. A group that's working on point of care diagnostics, again, where you could potentially at some point maybe have a bedside diagnostic versus having to send a test off. Vaccine development, there are 4 platforms here on campus within IDRN that are being explored. Better understanding of immune responses, as I mentioned before. Pathogenesis and animal model development. So are there animal systems that we could utilize to better understand the disease, again, for therapeutics and vaccine development downstream? Viral genomic evolution. So is the disease, the virus itself, mutating, and will that have impact downstream on infection rates? Serological assay development, better understanding of cardiovascular complications that have been occurring in individuals. Again, the impact of cancer comorbidities and other comorbidities. T-cell responses, so it's specific cells and how they interact with the human microbiota and whether that has an impact on the severity of the disease. Potential for maternal fetal immune responses that could have downstream impacts on babies. Respiratory immunotherapy, monoclonal antibody screening, again, developing tools that we can use for future assay development. Convalescent plasma therapeutics, sensor for detection of airborne COVID or SARS-CoV-2, and then companion animal impacts. What impacts might they have? And so one thing that I can say about these individuals that are part of this team is that they pivoted so quickly. They dropped their own research and started working on ways that they could address what was going on with COVID. They have true grit, tenacity, an incredibly collaborative group of individuals that are passionate about what they're doing, the science that they're doing, and their need to be a part of this, and very can do. This is a group that has taken a situation that needed to be responded to quickly, and they did it. So they've really risen to the challenge with their expertise to be addressing the COVID disease. So what is COVID? SARS-CoV-2, severe acute respiratory syndrome coronavirus 2. SARS-CoV-1 was the MERS disease that you may have heard of in Asia a couple years ago, but SARS-CoV-2 is what causes COVID. Here you can see a little depiction on the left side of your screen, a cartoon of what the disease looks like and how it binds to get into the host. And then another cartoon up here to the top with the red bulges. These are the crown components for coronavirus, is how it got its name, and then a couple of electromicrographs of the virus itself. So again, you can see these spikes here, or crowns in these diseases, or excuse me, in these virus particles. So epidemiology. Yeah.
Sorry, Candace.
Yeah.
Quick question from somebody. Is it okay if people can screenshot these slides and then share
Yes,
social media?
certainly. Yes, please, feel free. And I will also send the slides or a PDF of the slides to you as well. But yes, please feel free to.
Awesome.
Do I need to go back?
I don't think so.
Okay.
Yeah. And like she said too, after this, I will send out an email that has a survey, and I'll attach the PowerPoint with it too.
Great. Thanks, Corey. So where is the disease, and what do we know about the disease? So as we all know, the disease likely initiated in Wuhan in China, has now spread across the world. I pulled these numbers on the left-hand side of your screen about 15 minutes before we started. Global cases, we have about 24 million global cases of COVID, and these of course are the tested. Likely that's an underrepresentation of the real number of COVID cases, because not everyone is going to be tested. Same for global deaths, about 828,000 deaths. Here in the United States, we're getting close to 6 million cases here in the United States, with 180,000 deaths to date. How is the disease transmitted? And I know you have likely heard a lot about this. You know, over here on the left, it talks about an infected individual, and this infected individual could be showing signs of the disease or symptoms of the disease, or not showing symptoms of the disease, and can still be transmitting it through either droplets. It appears, like most things, smaller droplets can find their way further down into your respiratory tract than larger droplets. And so there's been recently this concern about airborne transmission of disease, direct contact, indirect contact, and of course a susceptible individual. And it appears we are all susceptible. This is a novel virus. We've not seen this virus in the past, and so that makes us all incredibly susceptible to the disease. Again, just a quick way that it finds its way into the respiratory tract, into these small pockets called alveoli within our respiratory tract. That's where the virus actually initially takes hold. The deeper into the respiratory tract that it goes, the more severe it appears the cases are. It interrupts our immune system, is really the process of that. So from upper respiratory versus lower respiratory, there's a larger immune response that occurs when it gets further down into your lungs. So we know that's also more than a respiratory virus. So the typical presentation of the disease, again, fever, dry cough, exhaustion, you're not eating. Really the number one symptom to date has been smell and taste disorder. So you lose your ability to smell and taste, myalgia, shortness of breath. But there are also presentations of nausea, diarrhea, sore throat, runny nose, and headache, and other cutaneous manifestations. I think you've probably heard about people that have had issues with their feet and the problems that come with that, as well as severe presentations and comorbidities increase those severe presentations of the disease. And those can include anything from cardiovascular disease, diabetes, hypertension, chronic lung illness, and kidney disease. Likely others that we're not aware of to date as well. So testing, how do we do testing? So there's a couple tests, well, there are several tests on the market right now, but there's two main ways of testing. So serum antibody testing, tests to determine whether or not you've had an exposure to the virus in the past. So you have produced antibodies. And there's a lot of information out there and a lot of controversy about how long those antibodies may actually protect you. And I was reading yesterday or day before about how the are now seeing some secondary infections that may be because the antibodies wane or go away with time. There are a couple other explanations for that that don't have to do with antibodies, that maybe the virus is just latently in your body and then finds its way back out again to infect you. Perhaps the virus is mutating. Some evidence is suggesting that small mutations are occurring that may cause these reinfections, but that it's way too early to really be definitive about what that is. So serum antibody testing right now to determine whether you've had a past exposure, and then of course determine whether you have an active infection using PCR antigen testing. And so what PCR does is really it takes a small little bit of the virus and it amplifies it to a large number of those viruses, to the nucleic acids within those, so that you can read it on a machine that tells you that there are multiple copies of that nucleic acid within that individual sample. And so to date, there have been the nasal swabs, and more recently have been saliva tests that they can determine whether or not you have a current infection. Of course, you've also heard a lot of information about how good are any of these tests and what can be improved upon them, and that's been a real problem over time. So what has been the response of IDRN, and what are they doing for COVID? They've been doing several things that, as I mentioned before, in that laundry list of things that folks have been working toward. I think one of the other pieces that they've been doing is they've been ensuring their infectious disease expertise. So we've had folks that have written pieces for The Conversation. We've had folks that are recreating a live animal market and trying to better understand how that disease was transmitted, so that we can share that information about maybe what not to do to try to mitigate the spread of diseases. These coronaviruses, this isn't going to be the last disease that can find its way. One of the areas of real interest within our department as well as the college is how reservoir hosts and diseases transmit from animals to humans. And it's a real issue that will continue to be. The forum, as I mentioned before, providing more local information spread to individuals both here on campus as well as the community at large. And then a lot of information, again, this goes back to the fact that we've had this expertise within our department, within our college, within our university, that can provide information because they've been studying things like bats and coronaviruses in bats. And can we better understand from what individuals have learned from other viruses and how they transmit from bats to other species, and utilize that for COVID and sharing that information. We've also been working on community solutions. I mentioned that a group has developed hand sanitizer here on campus, and then one of our graduates from CSU, an alum from the microbiology, immunology, and pathology department, has a brewery here in town. And so he reached out and have been collaborating with researchers on campus to make hand sanitizers in bulk so that it can be used and distributed to businesses here in Fort Collins as well as on campus. Engineers have been working really quickly to try to better understand disinfectants and what are the best disinfectants, and are there alternatives to the alcohol-based disinfectants that we've all been using. And then within our department, it was clear for us to be able to come on campus and to be able to do our research in any capacity, whether that would be COVID research or research that has been ongoing on campus, that we were going to need a few things, and one of those was masks. And so within our department, there were about 25 people that stepped forward and made over 500 masks for our department, and that sort of has grown outside of that in helping to distribute masks to make sure that people are safe within the laboratories when they come back to work. Testing, treatment, and transmission surveillance. So as I'd mentioned before, one of the very first projects that got underway was looking within the retirement communities here in Fort Collins. And not only to try to better understand what the transmission looked like and whether the healthcare providers were a part of this picture, but also to start looking at whether or not this virus is changing and mutating. And so that work is ongoing here, right here at CSU within the IDRN. There's another group that's been heavily involved in testing antiviral compounds. So compounds that already have FDA approval and can be put into a culture system pretty rapidly to try to determine whether or not it can kill or mitigate the virus. And so they've been doing some incredible work. They've been now collaborating with a variety of individuals and companies, both within CSU and outside of CSU, to try to find antiviral compounds that will are very important, especially prior to the development of a vaccine. So as I mentioned, the point of care or bedside testing, there's a group here on campus that have been working several years on the development of a point of care diagnostic. Again, could you take this, swipe it inside your mouth, and determine whether or not you had COVID or any other infectious disease right at that time versus having to send it out for analysis through the PCR analysis. And then there's been a lot of partnering that's been going on here in Fort Collins, as well as the state of Colorado. And so wastewater testing. So could we surveil wastewater to determine whether there's an active or a potential outbreak of COVID in an area? So if you look at different areas across campus or different areas around Fort Collins or other waste treatment plants, could you determine whether COVID is in that wastewater? And so we have a group that's been collaborating with a company here in town to do that. And then of course the Veterinary Diagnostic Laboratory is now processing human COVID-19 tests. And that has been a huge benefit for Colorado. And there's a great partnership that's developed between the Veterinary Diagnostic Laboratory here at CSU and the state of Colorado to try to improve testing outcomes. And I should say too, the surveillance and survey for students here on campus. So some of the surveillance work in development of antibody assays, serology assays, are helping to try to better understand what's happening on our campus population as well. And then of course, vaccine development. There are 4 vaccines that are currently part of IDRN that are being developed. And these are all come from platforms that already existed that they could put in COVID-19 into their systems. And one of them was a group of researchers that study tuberculosis. And they were looking at world maps and looking where prevalence rates of COVID were, and found that countries that typically vaccinate for tuberculosis had lower COVID prevalence in their countries. And so the question was, is there something about that platform that could be used then to address COVID-19 or SARS-CoV-2? Poxvirus also being used as a pathway to try to develop a COVID-19 vaccine. Again, someone here on campus who's already been working on that. Another researcher is who works in coronaviruses. He actually works in different types of cat coronaviruses versus the human coronavirus, looking at whether or not yogurt or lactobacillus could be a delivery method for a vaccine platform. And then the 4th vaccine is Solovac, so using riboflavin and photosensitivity to inactivate the virus. And again, something that's being used right now and developed for blood products, can we use that to try to mitigate the COVID-19 disease? So all the work that these individuals have been doing, and you'll be hearing more from many of these groups over the fall semester, more specific about the vaccines and about other approaches that are being done here on campus. Again, I'm just trying to give you a global sort of perspective of what's being done. But all of these groups have been so successful in what they're doing is that CSU was actually one of the top 10 best universities solving the coronavirus pandemic as determined by successfulstudent.org. We're actually number 4. It was very exciting to see that all the hard work that individuals are putting into their research and their service are actually reaping benefits. So what else is IDRN doing? So I talk about all these things that we're doing on the research platform and bringing students in and faculty in. Again, I am really interested in science communication and how can we be better communicators of our science, reach a larger group, and really instill new innovative behaviors and learning processes in students. And so I was very fortunate to attend the Alan Alda Center for Communicating Science last fall, in November actually, right before we really realized what was going on with the SARS-CoV-2 virus. And so I'm bringing that here to CSU. It was going to be here this summer, but unfortunately, those plans had to be rerouted. And so next summer, the goal is to have faculty, students, and staff coming together to facilitate their ability to tell their own science stories for the future. So what does the future hold for IDRN? Certainly we'll continue with the development of these IDTs. We're working on a T32 grant right now, which is a grant that would allow more students to become involved in the work that we're doing. Promoting the collaborative approaches, of course, involving students, faculty, and staff in this, the science communication angle. I always think it's great that we can learn and be better communicators of science, not only to the scientific community and how we share our work, but also how we share these stories and how we better present this information to the community at large. All of this to come up with more innovative techniques and ideas to become better problem solvers. Again, to investigate, study, and develop mitigation strategies for diseases, including COVID. So with that, I would be happy to take any questions that you might have.
First question is, if this type of presentation is any example, it's just a honest comment. You gotta, says you are an A1 at communicating your science to the community at large. So thank you for what you're doing.
Thank you. I appreciate hearing that.
And again, I know a couple other people popped in a little bit late, but if you have questions, feel free to put it in the Q&A box or in the chat, and we'd be happy to answer anything that you guys have right now.
Thank you, Carol. I see that came from you.
Gosh, if not, that means Candace, you did a great presentation. Nobody has any questions for you.
Okay. Well, thank you. Thank you for the opportunity to give just sort of an overview of what you'll be hearing the rest of the semester. I'm excited for all of you to be able to get in on the ground level of what people are doing here at CSU. They're doing an incredible job.
Oh, and we just got a question in on this. It says, you touched a little bit on T cells. Can you elaborate more on that?
Yeah, so yeah, yes and no. There are ongoing studies right now looking and using animal models here at CSU to try to better understand which cells are responsible for what immune responses that are occurring. And so I think right now it's too early to actually give much information. The hamster has become a really incredible model to be used for these studies. And there are two groups. There's a group that has been really looking at a variety of different animal models to see what we can learn and whether or not they're susceptible. And I honestly think, I don't know what, do you have Marcella Hyeno Tomeo speaking this fall?
I don't think so.
Oh, that's a bummer. So Marcella is actually the one that's doing this work, and she would be someone that you might want to follow closely in the future as to what's happening with specific cells. And I think it's really too early to really know what's going on.
Oh, we just got a couple more questions that just came into. Number one was, has your studies worked with students and others on campus as we start this semester? Or how have you guys been working with students too?
So bringing students in to do a research component We've had some actually there were undergraduate students. They graduated in spring, and they've been heavily involved in research, as have graduate students that were part of individuals' labs. And so students have been actively involved in what's going on, as well as I mentioned the students. Right now, students are being surveilled for COVID as they come on campus. And so that work is also, involving students, surveilling students, trying to get a better understanding of what the disease state looks like here on campus is another way. But yes, students are involved in the work. They've been trained in BSL-3 facilities. They've had incredible opportunities to learn and to grow, to be able to take this to the future. In fact, that one undergraduate student, she's now a graduate student here at CSU, got a science publication just in the last month from the work that she's been doing on some of the antivirals. So it's really exciting to see students be a part of this.
Awesome. Next question is, what would you say the biggest progress has been made through your team?
Yeah, gosh, so biggest progress. It's so hard to pick out one thing. I think one of the things that, it's hard to pick out one thing. The surveillance information and better understanding asymptomatic carrier state, that was a real turning point, I think, for not only CSU, but for the state of Colorado, and the response that the state of Colorado's had. They funded that work when we initially started doing that somewhere April-ish. And so it really gave them a better understanding of the asymptomatic status that was here in Colorado. And so it probably had a pretty big impact on how our governor led his response.
Awesome. What's your number one takeaway, the number one elevator talking point about COVID in CSU?
Oh, we're dynamite. I mean, I think it's the typical things that I say about individuals. COVID and CSU. This is such a caring community. People were coming out of the woodwork to want to do this work, to want to switch their research. Just a compassionate and collaborative group of individuals.
Yeah. If somebody is asymptomatic, are they not contagious after a 14-day quarantine?
Yeah, that's a good question. I think that's probably still open for debate, but right now the going thought is the answer to that is no, because they have done some testing across time. An asymptomatic person, or anyone actually, is most infectious right before they become symptomatic. I think the data, though, that was done here at CSU was showing that individuals that were in the asymptomatic category, they could have a high viral burden or a low viral burden. And so I think that probably will have some implication too as to how long you may be in that carrier state or able to transmit the disease. But I think the work that's been done has shown that between 10 and 14 days, that your viral titers go down considerably in that time.
Awesome. And then can you address a little bit more about what being asymptomatic means?
Oh, yeah, sorry. Asymptomatic means that you don't show any signs of the disease. And so you look healthy, you don't look like you're infected with anything, like me right now. I am not showing any signs of disease that I showed earlier. And so, but I still could have the disease if I hadn't been doing the proper things. And even then, I think there are people that don't know how they contracted COVID because they think they've been doing all the right things. So asymptomatic just means you're not showing any signs of the disease, and you don't even know yourself that you might be infected.
Awesome. And then what do you think is the biggest roadblock to solving the COVID-19 pandemic?
Oh gosh, there are many roadblocks. Solving the pandemic. So having a vaccine that is efficacious, that is sufficiently efficacious to mount a response, maintain that response, and then enough individuals being willing to be vaccinated with that vaccine. to maintain that. So the idea of getting to herd immunity, which herd immunity means that around 60 to 70% of individuals have the disease, have had the disease in some form. Doesn't necessarily have to be a real whopping disease, but enough so that you don't get the disease again. It has to be 60 to 70%. And we're probably now around 5 to 8% in areas that have had pretty bad outbreaks. And so the thought of getting to that anytime soon is problematic. So really vaccine development for long-term is probably our best bet. And then the development of antivirals, I can't stress how important that is, so that we have something that we can use to help people get to the other side of infection.
Awesome. And then worldwide, what would you say the progress is on a vaccine or a treatment?
Yeah, that's challenging to say. I think I heard yesterday there are like 170 or 180 vaccine trials that are out there right now. There are only a handful of those that are in what would be called third stage trials. And so, gosh, I don't know. It's going to be really challenging to try to find a vaccine that is going to be effective enough and to have enough people take that vaccine. Yeah, that's going to be a challenge.
And then let's see. Do we know that having the disease confers long enough term immunity to help towards herd immunity?
Yeah, that's been one of the other concerns. So just this week, people have started reporting. There were some questions as to whether or not people were being reinfected 2 or 3 weeks ago. But this week, there are more cases in Europe, in the Netherlands, in China, and even here in the United States, where people think that they are second infections. And so how do you establish a second infection from just the same infection that went dormant or latent in your body and then came back out again? And so I think one of the concerns is these are individuals that are about 3 to 4 months out from their initial infection when they are testing positive again for COVID. And so the question again being, really, will we be able to get protective immunity over time, or will we reinfect? And there's still not enough information out there. There's a small slice of evidence suggesting that these new viral infections are from a virus. It's the same virus, but has some mutations that allow you to become infected again. But I don't think there's sufficient data out there to tell you that that's really what's going on. So I think it's a real concern right now as to whether or not we'll be able to maintain long-lasting protection against the infection.
Awesome. And then will CSU take volunteers for a vaccine trial?
Well, that's a good question. I doubt that will be run through CSU. My guess is that would be purchased by a company, but I totally do not know the answer to that question. I kind of doubt it though.
Okay. And then can you talk about the safety of vaccines and the testing that is required for a successful vaccine to be approved?
Yeah, so you're going to get a big bang of this by the people that are talking to you about vaccines, I think, over the next couple of talks. But there are several things that have to happen. So it needs to be safe. It needs to be able to provide some protection, and it needs to maintain that protection over time. And depending upon what the platform is, there are many known platforms and there are many unknown platforms that are being used. And so some of them we already know, talk about, polio vaccine or smallpox vaccines or other vaccines that we have, the influenza vaccine, they use some of the same platforms. And so those would be thought to be a bit more safe than others because we know how the body responds to that. There are other vaccines that are out there right now that are using newer platforms that we either have never developed a vaccine that's been out for human, use, or we have very little data on those. And so That's why we have the trials. And I have to tell you, so I did some vaccine work many years ago, and it's really hard to develop a vaccine that can do what it needs to do. And then to be able to get that through animal studies and then through human studies to verify that it actually can work. And that can take anywhere from 5 to 10 to more years to do that. So the movement that we've done as a global attack on this has been pretty rapid, which has its pluses and its minuses in knowing whether or not it's safe. So I'm sure you've all heard about that you can sign up for some of these vaccines and be a test pilot for one of the vaccines. I think it's too early to know what those responses are going to be. I think they've had some protection, and I think the Oxford vaccine probably has the biggest leg up right now. But I think we're still in a wait and see situation.
Awesome. That's all of the questions that I have so far. I know it's 6:20, so we still have 10 minutes if anybody has more questions. People are definitely saying thank you for this. This has been huge and helpful, I know for myself as well. For everybody that's still on too, just to let you know, next month we will be talking about the Solavax vaccine specifically. And we have a couple of presenters that will come in and talk about the research that they're doing and the process on where it is on testing. And that one will be on September 22nd, same time, 5:30 to 6:30. Also, I will post in the comment or in the chat again for everybody to look at where you can find more information about what's going on as far as research and the different departments that are going on and all of that as well. While I was talking, we did have another question that came in. It says, how does the FDA determine what is safe?
Yeah, so it's a long process. Typically, it's a really long process where you start in cell culture, so looking at a plate in vitro, we call it, in glass. So you're looking at cells and whether or not the vaccine has any detrimental effects to the cells themselves. And then it typically moves into animal studies, so animals that are susceptible to whatever the infection is or a surrogate of that infection. And so then it has to go through those animal studies. And then you go through the tiers of human studies. And there are 3 tiers of human studies before it actually finds its way for FDA approval. So it can be a really long, laborious process in doing that. We have really skipped forward considerably.
Awesome. Well, on that note, thank you again, everybody, for making it. Candace, thank you so much for being the first person to do our speaker series, as well as leading the charge and doing such a great job. I know that I learned a lot. And
Thanks.
I hope everybody else that attended did too. And on that note, I will let everybody go. You guys got it off 8 minutes early. We will stay on until about 6:30, just in case other people have more questions. And thank you so much, and go Rams.
Thanks. Thanks to you all. I appreciate you being here this evening. And I really go to all the others because these people are so cool.
Awesome. And then Candace, while people are popping off, yeah, if you send me the PowerPoint and everything, I will. Put it in that survey and everything, that'll be great.
Yep, will do. Thanks, Corey.
Also, because I want to look more into it too.
Yeah, great. Thanks. Okay, talk to you later. Bye.
All right, talk to you later. Bye.